In this chapter, we now take an account of the second part, which is meiosis. Initially, we had already said that meiosis was more concerned, or it is more related to the reproductive part. So, when we talk about meiosis, we'll have to say that meiosis is reduction division producing gametes. Meiosis takes place in the reproductive organs like the testes and ovaries. When we are talking in terms of humans, we'll have to once again mention that the testes and ovaries produce sperm and ova. When we talk about the animal part or the human part, we'll also make a passing reference as far as the flowering plants are concerned. In case of flowering plants, the process of meiosis takes place in the anther and the carpel, because in the anther the pollen grains are produced, while in case of the carpel the ovaries are produced, which contains ovules, as we already know. We have already studied a part of that in our previous year. So, coming back to meiosis, we'll have to say that in meiosis, the chromosome number is half. In case of humans, we have got 23 pairs of chromosomes that are there. One member of each pair is haploid, which is passed on to the sex cell. So here we'll have to say that this is essential because the male and the female gametes which are there, when they fuse together, which are in an haploid condition, they now become diploid. We will take into account that the diploid number is always represented as 2n, while haploid is n. So, in case of meiosis, we'll have to say that haploid number after fusion becomes diploid. We now take into account the second part, which is meiosis. Meiosis is a highly complex process involving numerous stages. The meiosis is completed in two divisions. The first is reduction division, the second one is mitotic division. We'll take an account of the first meiotic division, which is called as reduction, or it is called as separation division. Let us see on the monitor the details of it. As you can see, the chromatin fibers condense into chromosome. Homologous chromosomes, that is, the matching ones received from the mother and the father, come together in a pair with similar genes opposite each other. Each chromosome by now has also split into two chromatids, just like in case of mitosis. Thus, each homologous pair is now showing 4 units. 4 chromatids, collectively called as tetrad. Tetra means 4. So at this stage, the chromatids that we were talking about is now referred to as tetrad. So the condition is tetrad condition. In meiosis, one of the most important thing that takes place is the chiasmata formation. During pairing, some chromatid of homologous chromosomes break and rejoin at points of intersections called as chiasmata. Singular is called as chiasma. This causes crossing over with exchange of chromatid material between chromosome from the parents. This leads to a new combination of genes in the sex cells that are there. The nuclear membrane disappears and chromosome moves apart, becomes separate, and new 2 cells are formed. So, a process of crossing over is called as a chiasmata formation. The point of intersection, please remember, is called as chiasma, and the plural of that is chiasmata. We also take into account the second meiotic division, which is nothing but the mitotic type of division. What happens in this? the two daughter cells, as you can see now, they undergo mitosis in which two chromatids of each chromosome separate and move apart to get surrounded by the nuclear membrane. Thus, 4 cells are formed. Each finally resulting cell has only half the number of chromosomes, that is, haploid number n, of the original, which was diploid, or 2n. So, in case of meiosis, we are talking in terms of the first part, which is the meiotic division 1, and the second part, which is the meiotic division 2. Along with that, we have also taken an account of the chiasmata formation, or it is called as a crossing over. Here, we complete the part of meiosis. After studying the entire part of meiosis, let us also account for the significance of meiosis, followed by the comparison between mitosis and meiosis. The significance of meiosis on the monitor for you. The first significance of meiosis is, the number is half in gametes, that is the sex cell, so that fertilization will result in the restoring of the normal number of chromosomes, that is, N-N fusion will result in 2N. That's point number 1. Point number 2 is, it provides for mixing up of genes which occurs in 2 ways. Let us see what the 2 ways are. 1. The maternal and the paternal chromosomes get mixed up during the first reduction division as they separate from the homologous pair. And second is, while these are separating, the chromatid material very often gets exchanged between the members of the homologous pair, which we studied under the heading that was called as chiasmata formation. So here, a variation can be seen. These were the significance that we have accounted for meiosis. But let us compare what is the differences that we find when we studied mitotic division and meiotic division, because such type of questions would definitely feature in our exam. So here on the monitor, the significance followed by mitosis and meiosis. The points are, mitosis is occurring in somatic cell, while meiosis is occurring in the reproductive cell. Mitosis occurs continuously to provide growth and replacement. Meiosis will result in gamete formation. In case of mitosis, 2 daughter cells are produced. In case of meiosis, 4 daughter cells are produced. The fourth point for comparison is, the full set of chromosomes are passed to each daughter cell in mitosis. In meiosis, half the number are passed. The fifth point is, chromosomes and genes in each daughter cells are identical. In case of meiosis, they are assorted and they are random. So there is a genetic variation. Please include these words: assorted, random, and genetic variations. So here are the points, as you can see on the monitor, for the differences between mitosis and meiosis. We complete our very first segment of this entire chapter, which is cell division. We'll be moving on to the second part, where we study details of chromosomes.